Absolutely, that's the third line of defense. But what is
It has memory.
there we go. That's the most important thing. It has the ability to keep memory. So the next time you're exposed to that specific antigen, you already have the defense available, and you will not get sick secondary to that. You're already going to be prepared. And then how you have the cells that are available for that are T cells and B cells. Literally, I just met the father of B cells this week. Like when I was talking to him, one of the doctors today, he's like, you know how important that guy was that you saw him in lecture the other day. I'm like, I gathered, because he's a member of all these really important scientific societies, and he's led all of the almost every important thing that we know about B cells. This guy's almost discovered it. So he's an old man now, but he's still teaching, he's still lecturing, he works at Emory University. So, kind of cool. Immunocompetence. So where are B cells and T cells made? Where are they made?
The bone marrow?
They're made in the bone marrow. So everybody starts out in the bone marrow. Like all of your cell lines start out in the bone marrow through hematopoiesis. So after they're made in the bone marrow, though, T cells and B cells absolutely have to go get taught. They have to go grow up. So they have to go to like finishing school. Think of immunocompetence as finishing school. They have to go learn what to kill and what not to kill. B cells learn that in the bone marrow. T cells learn that in the thymus. Right? So an easy way to remember that. So during that process, they get exposed to antigen. And if they don't respond to the antigen, they don't pass the grade. They get killed and recycled. If they respond to things that aren't antigens, like yourself, they start responding to cells that they shouldn't respond to. Those guys get killed too, and they get turned back over. A very small percentage of the T cells that go to the thymus for maturity actually make it through, actually graduate out. They have a little ceremony for them. They get little hats. I'm just kidding. So, and that's a good thing, because you want that to be right. Because a T cell that goes after you is called, what is that process called? Autoimmune disease, and we don't like those. So once they get mature, then they go to the spleen, where they will first encounter true antigens. Then they'll go to the lymph nodes to hang out for their first job. So they go to camp to become immunocompetent. They go to the spleen to get their first assignments, and then they go hang out in the lymph nodes to really start doing their work.
So how do we get autoimmune disease if they're trained?
Some of them go off track when they're out doing their job.
They go rogue?
They go rogue.
Is
That's after they, and sometimes what causes them to go rogue are things like stress, things like a viral infection. A viral infection can set that off. So sometimes you can have the genetics for a certain kind of disease. You have the phenotype for it. You have the genotype for it. But you won't necessarily, the phenotype may not express itself unless you have something that turns a button up. And sometimes that can be a virus, sometimes that can be stress, sometimes that can be lots of things. Sometimes it can be a surgery. Like it doesn't, we don't always know what makes the phenotype express itself. There is we're just out there, Epstein-Barr virus, mononucleosis, kissing disease, y'all have heard of that? We're learning now that has a close association with multiple sclerosis. Y'all know y'all heard of that disease?
Yeah.
Mm-hmm. Causes demyelination of the nerve axons, and you start to lose your ability to walk, talk, think, all the things. So that's those, like We're finding lots of pathways to that. And so they can even turn a T cell on to become a rogue T cell.
Is there any autoimmune disease that attacks directly on the thymus or red bone marrow, and instead of turning them rogue, just let the ones that were already bad, like, free or something?
Anything is possible. I don't know the direct answer to that, but once something goes bad, it can happen in the bone marrow, or it can happen to ones that are already out and about. T cells. There are 3 types of T cells we're going to learn about. You have cytotoxic T cells. Cyto means cell, toxic means clearly bad. So cytotoxic T cells, these are your killer cells. These are the cells that are going to go up to something that's been marked for death. T cells have to have something presented to them. They will not on their own go up and go, oh, I don't really like you, and kill you. They have to be presented with something and shown, hey, you're supposed to kill us, and it goes, oh, okay, I'll kill it now. So that's one of our protections against that. So this is the cell-mediated immunity because it's a cellular process. It kills the very same way as which cells that we already talked about?
Natural killer?
Natural killer cells. These guys do the same thing. The difference is a T cell will be able to clone itself and make lots of T cells with the same receptor sites. So the next time it runs into that same guy, that same antigen, it's going to have tons of millions of those T cells available to form an attack. Helper T cells, CD4 cells, these helper T cells help instigate both cellular immunity, which is T cell immunity, and humoral immunity, which is B cell immunity. It actually opens the gates for both. It actually holds each of those hands to help both get engaged in the process of our immune system. And we'll talk more about that. Suppressor T cells, these are the guys, if our T cells get out of control, these helper T cells will calm them down. If the T cells are starting to kill things that aren't supposed to kill, these Tregs, that's what we call them, Tregs will come along and start killing the cytotoxic T cells and shut them down if they start getting out of control. We actually are wanting to use these guys in transplant as maybe a way to induce tolerance of these transplanted organs, especially in kids. If you're an adult and you get a transplant and you're like 55 years old and you live to 65 years old, kind of win. If you're a kid and you're 10 years old and you get a transplant and you live to 20, it's not enough. We'd like you to live till way more, right? So messing with these cells is really the answer. B cells, formed in the bone marrow. They mature in the bone marrow. And these are your basis of humoral immunity. B cells will turn into plasma cells, which will make antibodies. That's huge. So humoral immunity, so the difference between B cells and T cells, B cells don't actually kill anything. They will mark something for death. And an antigen-antibody complex happens, it doesn't kill the antigen. It can disengage the antigen and make it ineffective, but it doesn't kill it. It marks it for death. And in marking it for death, that's where the T cell comes into play. The T cell or macrophage, or another cell like that, or a natural killer cell. But B cells don't kill them. It marks them for death. Okay? It's a very important point to understand. So the difference between cell-mediated and humoral immunity, you will need to know this. So this slide is important. Cell-mediated immunity is initiated by T cells. T cells kill the cells themselves. That's why it's cell-mediated immunity. T cells actually kill the cells themselves. That's done with the help of cytotoxic T cells and helper T cells. Cell-mediated immunity actually happens inside the cell. Those T cells are actually getting inside the cell to create apoptosis. Pop them. Virus is inside the cell, cancer cell, transplanted cell, it goes inside the infected cell. T cells require an antigen presenting cell. Like I said, a T cell is not going to go up to just any old cell and kill it. A T cell has to have a cell that's marked for death or has an antigen presenting to it. I talked about antigen presenting cells the other day, right? So if a macrophage has eaten something, blown it up, now it has just this little piece of what's left of that epitope of that antigen. It brings it out here and puts it on the surface of this cell. That's an antigen presenting cell. Now, that T cell is going to come along, grab that antigen presenting cell, and then go around and look for any more of those. And if it spots those, now it's going to kill. It has to be presented on an antigen presenting cell. That's the fail-safe. That's what prevents T cells from going after every cell in our body. T cells have to have an antigen presentation. Humoral does not. Humoral does not. Humoral is actually creating the APCs. Humoral is marking cells for death with that antigen and that antibody antigen complex. Outcome is cytolysis by the infected cell, by the cytotoxic T cell. The response to this is antibody secretion by the millions by a plasma cell, by the millions. I had my flu shot on Monday. My arm was sore as stink. Monday night, Tuesday morning, now it's fine. But now my immune system's done its job. Still a little sore. But now I've got antibody. My B cells have done their thing. And now they're all located in all my lymph nodes. So if somebody comes up to me and coughs in my face and they've got the flu, I might still catch it, but I'm going to have a heck of a lot of defenders already sitting there waiting to shoot that virus in case it's going to try to attack me. So, again, here's another picture of the difference between B and T cells. B cells go to the bone marrow, get mature. T cells go to the thymus to get mature. B cells and T cells go get mature after that. They will go to a lymph node. And then a B cell would be a humoral response. T cell will be a cell-mediated response. Cell-mediated is going to happen inside of a cell, will cause apoptosis and cellular death. Humoral is going to create antibodies and will mark things for death. It does not kill the invaders. It will create antibodies to help mark them for death. Antigens are any substance that create immune response. So is mismatched blood an antigen? Yeah, because it creates an immune response, right. Peanuts can be an antigen. For some people, super cold water can be an antigen because it creates an immune response. Anything that creates an immune response is what that is for that human. Peanuts, chocolate, I mean, I've heard Benadryl. The thing that you treat people for allergic reactions can cause an allergic reaction in some people. It's nuts. So,
Rude people, sometimes they're just a
rude people, yes, rude people can absolutely, they are antigenic substances, yes. So, on the surface of the antigen, there are epitopes. These are the things, it's all a lock and key. Everything's a lock and key. So for that particular antigen, whatever comes to disengage it has to have a matching surface that matches it to disengage it. So the antibody would have to, so this would be the antigen, and an antibody would have to look like that to disengage it. It's a lock and key mechanism. It may not be on all of them. It may just need, you may just need one to do the same thing, to turn it off. You might not need all of them. You might just need one to have that same effect. So each lymphocyte has a antigen receptor. And that's where, when we talk about why we have to be re-immunized for things, the receptors change because they mutate. Does that make a little bit more sense now? When they change the receptor, we don't have a key for that anymore. So now we have to get reintroduced to the antigen to make more antibodies to make a new key. They change the lock on us. So now we have to have another locksmith to come along. But unfortunately, during that process, we have to get sick again. We have to get sick from the invader, make more, or we get a vaccine, and maybe it's not a perfect fit, but it's enough. It's enough of a fit that it's enough that we can get enough of some protection, right? They won't be able to get all the way through the door. Does that make sense? So antibodies recognize and attach to the epitopes, and oftentimes you're going to be attaching to more than one. So you actually cause clumping of those epitopes. You actually get a little clumping from all of them. You're attacking to many of them at the same time. It's a shape-to-shape recognition. That's how it works. And also that's how it doesn't work. If those viruses or if those bacteria change their receptors. The COVID virus was attaching in our bodies on the ACE attachment in our lungs. And they also have to have a way in. And so that was their way in. If we had found a way to change that receptor or block that receptor, we would have maybe been able to avoid getting sick from it. And people were doing that. They're like, oh, if I take an ACE inhibitor, maybe I won't get the disease that wasn't in fact true. T cells on their own will not recognize antigens. They have to be presented with an antigen-presenting cell, or an APC. They will not be able to recognize it without that help. Why are you laughing at me?
You really want us to know that, don't you?
I do, a little bit.
Yeah, I can tell.
Because it's really important. It's a redundancy to help our system not have autoimmune disease. So remember that dendritic cell that we talked about last Tuesday? A dendritic cell is an APC. A macrophage is an APC. Phagocytes are often called professional APCs. Because as soon as they engulf something and chew it up and eat it, they're going to put that epitope right on the outside of their surface of their shell. So macrophages, dendritic cells, and B cells are professional APCs. Man, that could be a good test question. Maybe not stated quite that easily, right? So, in the moment you have cancer cells, that also becomes an APC. Because what happens when you have a cancer cell, that cell loses its name badge, and now it's presenting something that's not normal. And your T cells will recognize that. And we're going to talk about what this MHC2 is. I'm afraid to say anything else. You're going to laugh at me some more. It's okay though. I'm okay being laughed at. And so, in this particular instance, we'll look at this. So this bacterium is engulfed. It's lysozymed. This epitope is put out here on the surface of it. And this MHC2 cell is now going to, this is where helper T cells come into play. Helper T cells will be attracted to this, and T cells will then become in play. We're going to learn what this MHC is. Major histocompatibility complex. So I've already told y'all more than once about how we all have little name badges. So every nucleated cell in our body has class 1 on it. So all these are on the surface of every body cell. They're on every surface. It's very important that we have these. They have two functions, self-recognition and antigen presentation. So one part is for self and one part is for our immune system. Every cell has these. It's amazing how we learned all this. We have two classes, class 1 and class 2. Class 1 is on the surface of all nucleated cells. So which cells will they not be on?
Erythrocytes.
Red blood cells. Very good. Strong work. It's 100% correct. And so class 1 will be presented to cytotoxic T cells. That's be a cellular presence, that is part of your cellular immune system. Class 1 will be presented to cytotoxic T cells. Class 2 will be on the surface of antigen that will be your antigen antibody presentation will be on class 2. Macrophages, dendritic, and B cells. So these are your APCs. That's what class 2 will be. And a helper T cell would need to be involved. Class 2 is for APCs. Class 1 is just on the surface of any cell. And if it's on the surface of any cell, a cytotoxic T cell is going to be there. So let's look up here. So you have an antigen presenting cell. This is an infected cell. Class 1 molecule is out here. It's showing that epitope, that antigenic piece, that epitope is out here on the set. This is a bad cell. It's been damaged. We have this antigenic piece out on the surface. The T cell receptor matches it. It says, I got this. Now this cytotoxic T cell is going to come along, and now it's going to shoot lysozyme and granulozyme into this cell, pop it, and kill it. The killer has done its job, right? Now over here, this antigen presenting cell, which is a macrophage, it has a class 2. If it's a class 2, you absolutely need to know this. You should write it down. A class 2 will always be a helper T cell. Class 2's major histocompounded complex is going to be as a helper T cell will be what will be engaged with that. That antigenic fragment, that APC, a helper T cell will be involved in that. So it won't be the same process as being blown up. There's going to be steps in between. So this is an APC, and this is a cytotoxic T cell. So humoral immunity. Humoral is going to happen outside of the cell. This happened inside the cell. We got inside the cell here. Humoral, we're never going to get inside the cell. Yeah.
How erythrocytes are gonna protect themselves if they cannot show the?
Say it again.
How do erythrocytes?
Red blood cells.
Yeah. How do can
Red blood cells, they can actually carry some, and they can actually carry things. They're like mules a little bit. They'll carry something that's been infected. The piece they can carry like antigen and something that's been marked for death on its surface, and as they carry it through the spleen, macrophages will clean that off, and as they go through, so they'll just carry and get cleaned off and then come back around. So humoral antibody-mediated immunity. In my world, antibody-mediated immunity is actually much more difficult to treat. We fight cellular rejection and antibody-mediated rejection. This is when the body goes, you're definitely foreign, I'm making antibody to it, and it makes antibody by the millions. Does not directly destroy the antigen, it uses antibodies to mark it for destruction. It marks it for death. And multiple cells may be involved in killing it. So let's look at this. Let's look at this closely. So you have an antigen on the surface of the cell. You have this B cell receptor. Okay, you have an activated B cell here. This helper T cell is involved in both humoral and cellular immunity. That's going to come up multiple times. Helper T cells are responsible for both humoral and cellular immunity. And because, look what the receptor site is here. What is that receptor site? MHC2. It's on both B cells and APC cells. So MHC2, there's going to be a helper T cell. You get this cytokine release. Cytokine actually helps stimulate more immune response. That actually makes the T cell release some granules, and it activates that, which will then activate the B cell some more. So you get this kind of circular thing that happens. So we have this binding. We come over here. This activated B cell will now undergo mitosis. What does mitosis mean?
Some division without.
Cellular division. Right, we're replicating ourselves, right? And then we make a lot of clones. Most of the clones will differentiate into plasma cells. If they're going to differentiate into plasma cells, what are they going to end up making? No.
Antibodies.
Antibodies. You're responding to the antigens. Antigens are what's producing the immune response. If they're turning into plasma cells, they're going to make specific antibodies to whatever that specific antigen is. They can do it to the millions. They will mark them for destruction. They will not kill them. They will mark them for destruction. What the clones that do not become plasma cells become memory B cells, and these can live in your body for years. Years. If you're healthy, if you have good nutrition, if you sleep well, if you're hydrated, those memory B cells can live in your body for years. This is your primary immune response. This is what the primary immune response means. This is the first time you've seen that infective agent and you've gotten sick by it. You're feeling this. You've got a temperature. You feel like a truck ran over you. You have myalgias. You know what myalgias mean? Strong word, yes. You have muscle aches. Your mouth is dry. You might be nauseous. You don't want to eat. Netflix is on your TV. You're curled up in a little ball. You might call your mom. You know, you're whining a little bit. This is your primary immune response. Now, you have plasma cells and you have memory cells. Now, I really want you to understand this. I'm going to explain it to you, but I want you to be writing stuff down. That first exposure, you create something called IgM, immunoglobulin M. And you get this little bump to that. That's your first exposure. That's your body having that primary immune response. But in response to that, those antibodies that are being made, now your second exposure, you're being, so you had this little bump, and now you've created something called IgG. It's floating around. And the IgG you keep in your plasma. Remember we talked about that way at the beginning. You have in your plasma that you have immunoglobulins, IgG, IgA, IgM. Immunoglobulins. IgG is antibody that has been created in response to an infection. So the second exposure to an antigen, all of a sudden that IgG gets turned on. Those plasma cells and those memory B cells, the moment you're exposed to that antigen, turn it up and start dumping those antibodies out in your bloodstream. And it jumps up. You'll never know you're sick. You'll never know you're sick. So you have these pre-made antibodies just rolling around waiting to find something to kill. Not kill, to mark for death.
Wait,
So
the first one produces IgM?
So the first time you're exposed is IgM, the rest, and then you make IgG for memory. Your first exposure is IgM, that's when you have your acute phase is IgM, your memory phase is IgG.
So if, for example, I have like some symptoms and I try for a disease and I find out that I have IgM but I don't have IgG,
then you have a problem making antibody and you have an active infection.
okay, so it's active, but what if like if I only have IgG but I don't have IgM? Does it mean that I was exposed but now I'm not sick anymore?
It means correct. It means you've had antibody but you're no longer sick with it. So let's use this as an example. Let's talk about COVID. When we didn't have a vaccine for COVID, they were asking for people who have had COVID to do what?
Give blood.
Give blood. Correct. So when they were asking for people to give blood from COVID, they didn't want their red blood cells. What did they want?
Antibodies.
They wanted their plasma and their antibodies. That's 100% correct. So they would spin that blood down, they would take their red blood cells away, they would get their plasma, and they would pull out, in particular, their IgG. They wanted their COVID IgG. They wanted the COVID antibodies that that person had made while they were, of course they were doing this with the people who survived COVID. The people who didn't survive COVID, they didn't want that.
Yeah, I don't think that was gonna take place
That
for
wouldn't
them
work.
anyways.
So they take that IgG, they purify it, and then they give it to people who are really struggling with COVID and give them antibodies to try to fight it. That process, that little bit of, that amount of IgG that they would give those people would cost upwards of $50,000.
The monoclonal?
Yeah, no, that's different. Monoclonals are different. That IgG that they were giving those people would be upwards of $50,000 for one treatment.
Did it work?
In the United States.
It probably made, it depends on how sick they were. For some people, it should have helped. If they had had a decent nutrition level, had a decent immune system, it should have helped. For some people,
But they
if
wouldn't really need
if
it,
they were
not
in
cancer.
multi-organ failure and they were throwing that at them at the end as like a Hail Mary, it probably wouldn't work.
But if they're like a relatively healthy person, like why would they really need that?
Because
in the
that's
first
what
place?
COVID did. COVID didn't care if you were healthy or not. COVID would go after your lungs. So it could stop the progression of really bad disease. So in that particular instance, it could help. So IgM, initial disease, primary disease. IgG is your secondary response. Absolutely need to know that. Memory cells can survive for some diseases up to 20 years or more. And that's true. Again, if I go check my varicella titer right now, what's varicella? Anybody know? Chickenpox. Chickenpox, that's varicella. If I go check my varicella titer right now, I will still have immunity to chickenpox. And I had chickenpox when I was 6. I'm not anywhere close to that. It's interesting. Basic structure of an antibody, you have a heavy chain that does not change. This will stay the same, and these light changes will become mobile and they will alter according to what they need to match the shape of the antigen. You have 2 of these sites. And they also have complement binding sites. What's the complement for?
Enhance.
Huh?
To enhance it?
To enhance it, from that complement cascade that we talked about earlier, right? So the complement, like the C1 to C3 to C5. So the same thing we're about to talk about. Complement, because complement had a couple of different pathways, right? Anybody remember the pathways of complement? Had a classical pathway, and the classical pathway was antigen antibody pathway, right? So you're about to see a lot of things that look familiar to what we talked about Tuesday night. Are y'all reading the book before y'all come to class?
No.
It will help you. Nodding head back there that said the test was easy or not so bad. There's a reason. Okay, thank you.
I don't know how to read.
Huh?
I don't know how.
To read? I'm sure there's an audiobook for it.
Okay.
If I read after class, that makes more sense to me than before.
Okay, read it twice then. Read it before you come to
I'm not
class.
gonna be able to pay attention.
You can
I'm
keep
for
making
anything.
excuses all you want.
I know my methods of learning.
Okay. All right, so these look very familiar, don't they?
Yeah.
From Tuesday night, because this was part of that complement cascade that we were talking about. So agglutination, when did agglutination happen?
Antigen
When?
response.
When?
Antigen response?
But when?
When it fails.
Is that the
Are you
first?
saying blood? So agglutination happened when we had blood mismatch, right? That was the only time we really ever talked about agglutination, when we had blood mismatch and it caused clumping. So, but opsonization, that definitely happened in the cascade, right? So let's talk about it up here. Pathogen coated with an antibody prevented from attaching to a cell. So just having the antigen antibody happening, it's blocking it. So if you have this antigen antibody complex, it blocks that virus, and that virus cannot get into any more cells, and it will not replicate because the antigen is blocking it from doing its thing. The clumping action, that is agglutination. That's what I was talking about earlier. When you have multiple antibodies that are attaching it to multiple antigens, that causes like an agglutination thing. So you're clumping it up, becomes very ineffective, can't get any more cells, can't start to divide. You've created a problem with it. And so that's kind of a clumping action. Neutralization of toxins. Toxins really happen a lot with bacteria. When bacteria die, it is very known that bacteria like to die and go out with a little bit of a bang. And so they'll release toxins. Viruses, not so much. Viruses don't release toxins. Bacteria really do. But what else are we exposed to that has toxins? Animals, insects. Snake bites, insects has toxins. Some of those toxins have to be neutralized by antibodies. That's what some people are more allergic to ant bites than others, right? Some people can have an ant bite, doesn't do anything. Other people have an ant bite and it almost puts them in the hospital. That's an immune response to that. Bee sting's the same thing. Some people can have a bee sting, doesn't do anything to them. Other people have a bee sting and they go into anaphylactic shock, right? So, help with phagocytosis, so it coats the cells, opsonization, and then just the fact that you have an antigen antibody complex, you activate complement, which then complement starts all those cascades that we've talked about. Opsonization, membrane attack complex. Remember those we talked about on Tuesday night. So here's the classes of antibodies and their function. You'll need to know these. IgA. This is found in mucus, tears, saliva, and milk. It's definitely passed over from mom to baby. I'm a big supporter of breastfeeding. I understand that every mom can't, but breast milk is wonderful for babies. Lots of protection in there for babies, and also the milk will adjust. If the baby has an infection, mom's body will recognize that and adjust the milk accordingly for the baby. That's how amazing that is.
Yeah,
You're not going to get that from a bottle of Enfamil from the store. So there's lots, and this prevents pathogens from adhering and penetrating membranes. Now, that's to a degree. It can be overwhelmed. If the viral load of whatever someone just coughed in your face is so great, that's going to be quickly overwhelmed. They're going to get over your defenses. They're going to climb the wall and get over it, right? IgD is part of your B cell antigen receptor, functions on B cell activation. We don't talk about that one a lot. IgE, eosinophils. IgE, eosinophils. Think about that one that way. We've talked about eosinophils. What do eosinophils do?
allergies and parasites.
And parasites? Imagine that. Attached to eosinophils, parasitic infections, and allergic reactions. So if you're having an allergic reaction to a bee sting, your IgE is going to go through the roof. So, and your along with your eosinophils. So IgE is in response to that. So if you're having an allergic reaction, your immune response will have, and you will have extra IgE because of that. IgG constitutes 80% of your circulating antibodies. So this is your antibodies that you've made. If you're vaccinated, all the things that you're vaccinated to is in your circulating IgG. If you caught a cold this year, anybody had a cold this year? So that cold is probably either rhinovirus, enterovirus, human metapneumovirus, RSV, any number of those viruses. You are currently carrying around antibody to those viruses in your circulating antibodies. Those will tend to fade because you're likely going to catch a cold again next year, right? Maybe a different one. Maybe the one you catch next year won't be the same version of the one you caught this year because you're going to hold that antibody for a while. It's a really cool, interesting thing, right? IgM is what is secreted during what?
Initial response.
Primary infection. But this is the one that is around forever. When you give an IVIG, when you give blood, we will separate the blood. Whenever you give blood, they immediately separate it. They give the red blood cells over here, because we give that to just people who need red blood cells. We take the frozen plasma, we separate that out. And then we take the IgG out for everybody.
So you cannot find the IgA count with blood testing?
Say it again.
You cannot find IgA count with blood testing?
We can test for all the immunoglobulins in blood testing.
But isn't IgA only in mucus, tears, saliva, and milk?
But you can measure it. So if you are someone who's immune suppressed, you might need someone's borrowed immunity if you're immune suppressed. Intentionally immune suppressed kids or cancer patients or whatever, we will give them some IVIG to help them have some borrowed immunity. So an interesting thing happened, and another reason I know masks work. So during COVID, everyone was wearing masks. People didn't have a lot of colds. Colds weren't being passed around. So during that stretch of time, because colds weren't being passed around, those antibodies weren't being put into that IVIG. So typically, my kids that I have on IVIG could be the healthiest people in their household. The whole household could be getting a cold, and those kids, because they're getting IVIG, and they were like wearing a shield. They could walk around and ding, 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 things were bounding off them because they wouldn't get sick because they already had the antibodies to those viruses. After COVID, they were getting all of these colds, and they would get really sick from these colds. And we would come in and swab them, and they would have rhinovirus, they would have rhinoenterovirus, they would have human metapneumovirus. And it occurred to me, I'm like, oh, it's because the mask worked. Those were still viruses too, but people were social distancing. They were using masks, and so if they had a cold, they weren't sharing it with anybody. And so that IgG, they didn't have any antibodies, and it wasn't in that IVIG. That's interesting, right? So now that everyone's back and the colds are going around, and that's why these colds are a little worse than we've had, and it felt like these colds were worse, right? Common colds usually don't make you feel like you've been punted. But this year, colds are really taking people out, right? People were feeling really crappy from these colds because we hadn't been around them for a little bit. And they hammered us. So now we're going to have some more antibodies in our system. That's what it was. We didn't have any pre-made antibodies to even a little bit of those colds until they took us out of the knees. So you can question was it the right thing to keep everybody at distance to try to stop COVID? Yes. If we had actually done it, if we had actually truly done it and truly stayed away from each other and prevented the vector from passing, we could have stopped COVID in its track. But we didn't do it. And then we kind of half did it. And in the half doing it, we created all this other problem. But we've had some other issues from it. It's interesting stuff. It's just interesting things to think about. This is a very important slide. I actually really like this slide. There's going to be test questions about it. Naturally acquired passive immunity. So passive immunity means your body's done nothing to create it. It's immunity that you've acquired, pre-made immunity that you've just earned passively. Breast milk and through the placenta. This is mom doing her work. So immunity acquired from antibodies passed through breast milk and the placenta. How long is it recommended for moms to breastfeed after birth?
Usually it's like 5 years, right?
God, no.
Isn't it so?
No, but like supposedly
No, when they have
longer.
teeth, stop.
They're just standing there. So,
Yeah. 6 months to a year.
yeah, like
6 months to a year.
13.
And if some people can even just get through 3 months, that's good. But 6 months is tolerable. A year is great.
My nutrition teacher said like the longer you can breastfeed, like the better.
Yeah, well,
it
yeah.
is.
I'm going to tell you, 5 years is, so first of all, what it does to the woman's body is extraordinary. That baby is taking your nutrition. Women can have osteoporosis from breastfeeding for a year. For 5 years, forget it, she's probably going to have significant issues. So It's not, that's what I say when I say, if you can, you should, but you have to balance out what it's doing to mom. Because mom still has to take care of the baby after they're there.
That was never brought up. It was just about the
Interesting,
baby.
isn't it?
I had twins and I breastfed them, and I had to stop breastfeeding them because they were draining me.
Oh, yeah.
I could not keep up.
Yeah. And you have to alternate breast.
Yeah.
And you have to breastfeed. If you don't go to one side or the other, you end up with mastitis, which is the worst thing in the whole world. I've never given birth to anything except bad ideas, but I've been doing this for long enough to know that's a very bad thing. And even if you have an infection in the breast, you breastfeed through it. You don't stop. But the woman's nutrition is very much impacted through what she's doing. It's really good for the baby if the woman is keeping up. So here's the thing. You're the sole source of food for that baby at any time of the day or night. If you can do it for 6 months, that's great. If you can do it for a year, you're a superwoman. There's no way you can do that. There's 5 years is too much. By a year, hopefully you're on the table foods anyway. So you're supplementing even by a year. You should be getting onto whole milk and doing other things anyway. But the benefits to the baby are extraordinary. The milk that comes from you is set up for the GI tract. The babies love it. If you have a boy, the milk will be different than if you have a girl. That's the amazing thing.
But if you have twins and
same
one
thing.
of them's a boy, one of them's a girl.
Well, I don't know about that. That's an interesting point. I hadn't thought about that, but I'm assuming. But breast milk is amazing. But you have to take into account the health and well-being of the mother at all points during that process, including your mental health. Because one thing moms don't do is sleep.
Yeah. And also for the baby, like I feel like breastfeeding creates
A bond.
A bonus, depending on the mother also. So if you're doing that for like more than 2 or 3 years.
And dads can get in
that's
on
a
that
lot.
too. So they have.
How we gonna get in that?
So moms can pump and dads can feed. And actually they have these things where it's hilarious actually. It actually looks like a boob and it feeds, and dads can hold the baby and do skin to skin and feed. And it's oxytocin producing, it's bonding, it's a beautiful thing. I think there's a lot of benefits from it, but bonding with your baby is super important. Because if you don't, being a parent is the hardest thing you will ever do in your life. When you're ready to do it, be convicted to it, because it's the hardest thing you'll ever do.
Very so.
Okay, so that's passive, naturally acquired. Artificially acquired. This is what I was talking about with the IVIG. So another example from this would be, anybody heard of rabies?
Yep.
So if you want to go play with a raccoon in your backyard, why, I don't know, you'd want to do that. But let's say you'd want to go play with a raccoon in your backyard. You think it's cute, you scratch it, and it bites you. Well, now you are going to go get some rabies shots for fun at your local emergency room for a series of maybe 5 to 6 of these guys. What you are getting is rabies IgG. So they use this. I don't know if they are using an animal as a vector for this or not. But so they take antibodies from something that has had rabies. This is what they use for animals too. So they use, if animals get rabies, they just go to animal heaven. But if you get rabies, or you get exposed to rabies, you get something called, by the way, if you get rabies and you do not get treated for it, by the time you start showing symptoms, it's too late, you will die, period. Rabies kills people still in this country and in Florida. It's a deadly disease, it's nothing to joke about. So if you get exposed to it, you start this series of shots, and it's rabies IgG, it's rabies antibody. They give you shots in your stomach, or now IV, and you will get, it's actually IM now, and you will get a series of those shots. And you will basically get a dog tag. You can't, actually, I know people who have. But that's harvested from another person, it's artificial, It didn't come from another person. It is artificially acquired, and it's passive immunity. Your body did nothing to make it. It's artificially acquired. You have antibody running around your body that you did nothing to make.
For snake bites and stuff like the antibiotics.
Yeah, snake bites are the same thing. So if you get anti-venom, that is the same thing.
That IgG.
IgG. Active. Active, naturally acquired, is you got the disease. So people are not, for some reason, feeling overly moved to get their children fully vaccinated anymore. And it's people who have higher levels of education. I would argue that. Measles, mumps, and rubella. So mumps can cause encephalitis. It can sterilize males in particular. So if you get mumps, and we'll talk about this, it goes after the parotid glands. It goes into the scrotum of boys. If it gets into the scrotum of boys, guaranteed you will not give birth to anything. So if you get it though, you are for sure, and you survive it, you are for sure going to make antibody to it, and you will never catch it again. That's a guarantee. So you got the disease, you recovered, and now you're going to have active, you're going to naturally acquired immunity to the disease for the rest of your life. Never going to get it again. As long as your immune system is very strong. There are people that I know that I've heard who've had chickenpox twice. I think that's crazy. Other thing that can happen from something like chickenpox, people who get varicella can end up getting something called shingles later in life. So varicella is it's a latent disease, and then shingles can come out later. Y'all have heard of shingles? Your old people will talk about that because it comes out and it's very painful.
They just have like commercials about it.
Yeah, they have, because we're the only country in the world that allows commercials for drugs, by the way. But yes, yes, they have commercials for the shingles vaccine. Shingles hurts, causes a lot of pain, and there is vaccines for it because it's a virus that we can fight. And then, last but not least, artificially acquired active. So what that means is you've had a vaccine. It's an attenuated virus. Anybody know how many live viruses we do now?
How many what?
Live viruses. There are some live virus vaccines. MMR is a live virus vaccine. Rotavirus is a live virus vaccine. Varicella is a live virus vaccine. Everything else, we used to do polio as a live virus vaccine, but some kids were getting polio, so we stopped doing that. So now it's an attenuated dead virus. It's oral now. So if it goes in your mouth, what happens to it when it hits your stomach?
Die.
The acid.
And it gets chewed up. So those are the only ones that are alive. Everything else now is dead. So you get a shot, but it has enough antigenic properties that you're going to have a response to it. So you're required. Now I will tell you, this immune response is going to be much more solid response than this immune response. If you got the disease and you fought it and you survived it, definitely going to have a stronger immune response than this. But there's a little bit more risk with this one. You mitigate some of the risk with this. You're okay with risk? Okay. The other thing, well, no, I'm not going to get into it. Okay. Someone tell me.
D.
What did you say?
D.
As in dog?
Yeah.
Yep, that's correct. Everybody agree? Opsonization.
C.
C.
C as in cat? That's correct. Humoral? Does a humoral happen inside the cell or outside the cell?
Outside.
Does it kill? Or
No.
what's it do?
Marks.
Yes.
See, you were a great teacher.
That's right.
Artificial passive.
Artificial passive immunity. That's right. All right, cell-mediated immunity. Y'all are listening. I'm proud of you guys. Thank you. So, cell-mediated immunity responds to abnormal cells or pathogens within a cell. What's happening within the cell? That hurts. So, it uses 3 classes of cells to do this. Cytotoxic T cells, these carry out the attack. These are your attack modes. This is your Jason Statham's, whoever else you really like that's really like in the fighting kind of movies. Helper T cells, they activate the cytotoxic T cells. The same way they activated the B cells, they're going to do the same thing with the T cells. And the memory T cells, these keep memory of that pathogen in case they run across it again. Then your T cells are going to come out and do the karate chop again. So same thing, you have an epitope. You have to have an antigen presenting cell with T cells. So it's presented as an MHC2 cell. If it's the helper cell, if it's a T cell, you're going to have an MHC2 cell. It's recognized as abnormal. The T cell is activated. Once you get that T cell activated, then you have the same thing. You have that cytokine release. That cytokine release turns on the T cell. The T cell starts to replicate. You have 2 clone cells. That clone cell, you can also, at the same time, if a helper T cell is involved, you're going to activate actually both lines. You're going to activate a humoral and a T cell at the same time. T cells aren't going to your helper T cells aren't just going to pick a side and go. It's going to be open season. It'll get both your B cells and your T cells going at the same time. CD8 cells become cytotoxic, and then they will create that'll be sensitized just to that antigen, and then you'll create some memory cells from that too.
By on the other side, when you say in response to cytokinesis,
Say it again.
Cytokines. What those are the.
And there are chemicals that release from the cell. They're like chemical messengers that help initiate the cycle.
From both, or from just the T cell, the helper T cell?
From the helper T cells. Helper T cells actually carry cytokines within them. So when they attach to that MHC2 receptor, you release the cytokines. The cytokines get released from both directions. You get cytokine storm kind of everywhere. And so after you bind, after it binds, now you start to have the perforin. So perforin does exactly what it sounds like. It perforates the cell wall. Then you release lysozyme and breaks down, it lyses the cell. These are the cells that kill. So for the T cell to attach, it has to have that MHC1. So the cytotoxic T cell has to have that MHC1 receptor site. MHC2 is only for the helper T cell. But once that MHC1 site is there, the T cell will attack. So that has to be an antigen presenting. It has to be presented for a T cell to attack it. It has to be an APC cell. So an APC cell is presented to that cytotoxic T cell. It attaches, and now it goes into full attack mode, blows up the cell, and moves on, looks for the next one to kill. So helper T cell, this is important, and you'll see this. This is actually a nice slide that shows you what the helper T cell does in both. So there's an APC macrophage, so that is an MHC2. That protein there has to be an MHC2. And now you have, so I don't want you to get confused with these interleukins. So these are just different proteins that are involved in both phases of this, but you don't need to know these. But these interleukins are actually the name of some of the cytokines. So interleukin is responsible for the messengers that go back and forth. But cytotoxic T cell is going to be involved in cell mediated, and also you're now going to send the same messengers down here for B cells. So the whole point of this slide is, again, what I said earlier, when a helper T cell is involved in an immune response, you're going to actually get both cell lines going. You're going to get cellular immunity going and humoral immunity going at both times. Helper T cells can't pick a side and I'm just going to do cellular this time. You guys just sit on the bench. When it starts releasing cytokines, it's going to wake up at both sides of the immune response. Make sense?
B.
Yes, helper T cells, that's right, strong work.
D.
D, I agree. Everybody else agree? Definitely D.
C.
What?
C is in cat.
C is in cat? I agree with that too. That one should be an easy one. I've jumped up and down about this one. Cytotoxic T cells and natural killer cells kill the same. The only difference is what?
Memory.
Saturated T cells don't have memory. Well, not-
Have memory. So this is part of what immune system?
Innate immunity.
Innate, and this is part of the,
Adaptive.
yes, very good.